Hello, everybody. This is Serge Ferrari speaking from Geneva. Welcome on this new webinar from our webinar series of Bone Key and the IBMS. And our theme tonight is on the use of stem cells for bone repair and regeneration. The presentation tonight will be performed by Professor William Hill from GHSU in Georgia. Dr. Hill is an associate professor, PhD by training, and he is actually someone that comes from a totally different world than the bone world because by training and first interest, he comes from the neurological sciences. His interest for many years has been to investigate stem cells to repair damage in the brain following stroke. And from his experience in that field, he then moved into the bone field to try to apply some of the principles of tissue repair by stem cells to the bone. And he is now actually part of both the Brain and Behavior Discovery Institute at GHSU and the Department of Orthopedics, actually a part of a new institute of regenerative and reparative medicine at that university. So it's going to be a great pleasure for us to listen first to his presentation on this topic, and then as usual on our webinars, this will be followed by a discussion that will be held tonight by a very prestigious and a varied panel of experts, with both experts from the biological field, from the bioengineering field, and from the clinical field. And this panel will be composed of Dr. Céline Colneau from the INSERM in Paris, France, Dr. Mustafa Kassem from Denmark, Dr. Sandeep Khosla from Mayo Clinic, Dr. McDevitt from the Georgia Institute of Technology and Emory, Dr. Pioletti, my colleague from Lausanne in Switzerland. So without further ado, I would like to give the stage to Professor Hill to speak to us about the use of stem cells for bone repair and regeneration, dream or reality. Please, William.
Thank you. And I'd like to thank Serge for inviting me to give this presentation and for this very distinguished panel who've agreed to participate today. So this is an outline of what I'm going to cover today, looking at the status of stem cell therapies as they are currently in place, major challenges both in terms of institutional infrastructure, scientific and regulatory, which we'll talk about down here. What disease injury targets should we be looking at? We should be looking at late, early targets, easy versus hard, what's most common, what would be potentially the most profitable in terms of driving industry. What cells and cell sources, big question, allogenic versus autologous. Where should the cells be coming from in terms of tissue-specific origin? Matrices, the role in tissue engineering and modification of the cells and the microenvironment the cells reside in. And then how will various regulations affect some of these choices? So in terms of the current status of stem cells and what's in the pipeline, obviously there are thousands of investigators doing preclinical stem cell studies around the world. And in the U.S., as you move towards a translation, you need to run things through the U.S. Food and Drug Administration. And right now they've got over 1,000 investigational new drug applications, INDs, or investigational device exemptions, IDEs. And this is what you need to move a stem cell therapy into clinical trials. And then once you've gone through clinical trials and need to go to market, you need to go through a market approval process, either a biological license application or a pre-marketing approval. And right now, out of these 1,000 plus new investigational applications, INDs and IDEs in the BLAs and PMAs, it's not clear how many are bone focused right now, but there are many that are in the pipeline. If you look at the clinical trial databases, there's about 300 clinical trials worldwide that are looking at stem cell therapies based on bone marrow-derived stem cells or adipose-derived stem cells. In 51 of these trials, 7 in the U.S., are using stem cells focused on bone repair or bone diseases. And in this instance, they're almost entirely mesenchymal stem cells, either derived from bone marrow or from adipose tissue. And right now, there are no clinical trials focused on bone repair or bone diseases using IPS cells or embryonic stem cells. Now, if you look at sort of distribution of disorders that are in clinical trial, bone fractures, bone fusion, in this case, 4 out of the 5 clinical trials are in the U.S. I've highlighted that in blue. Bone sister voids, osteogenesis, and perfecta, again here in the U.S., 3 out of the 4 worldwide clinical trials are on this. Osteonecrosis, osteoarthritis, or cartilage repair. So to date, the FDA has not approved any stem cell therapy except for Hemacord, which is a allogenic umbilical cord derived product for hematological purposes. So there are no FDA-approved stem cell therapies related to orthopedic issues. In the private conversations with the FDA, there's a real concern on their part that there are no breakthrough therapies that are in the current pipeline, either in late stage, in the pre-marketing approvals, or the INDs and IDEs. And this includes bone-related therapies. So the FDA believes that most of the applications are for therapies that are only going to make modest incremental impacts or have marginal benefits for patients beyond current therapies. And this is a big problem. In contrast, worldwide there are hundreds of unregulated private and clinics that are sometimes related with government or military groups within those countries that are offering stem cell therapies with claims of very effective treatments and cures. So there's a big mismatch here. There's a real drive and desire by patients worldwide to have access to stem cell therapy, but the reality of what's out there in the near term of well-regulated and effective therapies is very minimal at this point. So the therapies that are available in these private clinics are various for many different disorders, but they also include very commonly treatments for bone, musculoskeletal, and joint disorders. Several in the U.S. are very active, and these are mainly using autologous bone marrow-derived or adipose tissue-derived mesenchymal stem cells. And so there's a real potential for these clinics to interfere with the legitimate development of orthopedic and other types of stem cell therapies, whereas there's going to be, again, this mismatch between the public perception of these therapies that are unproven, the effectiveness is unknown, and how that's going to contrast with what's available through very well-regulated techniques. So in talking to investigators at international meetings for tissue engineering and regenerative medicine, a number of issues keep coming up as major challenges for stem cell use and movement from the bed to the bench to the bedside. There's issues related to institutional infrastructure, and this includes research funding. So in the U.S., there's a very large political issue in terms of the use of embryonic stem cells, and this is true for a number of European countries as well. But worldwide, there's less of an ethics issue than in the U.S. So funding for this and regulation of embryonic stem cell use is very different and problematic. Investigators are also concerned that at the level of NIH funding, that the study sections that are reviewing research applications really lack support for translational research. They're focused more on molecular biological and cell signaling issues, basic science mechanistic approaches, rather than how can we take some of these ideas and move them forward translationally. And along with this, the NIH does not see that large animals are innovative, that the use of large animals as you transition from a small animal model like a rodent to a large animal, which is going to be very critical in terms of real-world biomechanics that a human patient might face, as well as immune systems that are more like that seen in humans. And this is a critical bottleneck or gap where the study sections don't see this as innovative and don't want to fund movement from small animal models to large animal models. And these are areas that I think need to be corrected in the U.S. funding stream. In terms of other funding, it's hard to get venture capital funding since they're looking for quick turnaround, and I think realistically to get something to market that's going to be proven effective and innovative is going to take a long period of time and take a lot of money. Big Pharma is obviously an area of interest. They are partnering with academics, but a lot of them are taking wait-and-see approaches on things. Philanthropy is limited. I'm going to talk more about scientific issues in just a few minutes, but this includes cell sourcing, expansion of cells to usable numbers, the use of matrices, and engraftment. And there are various barriers or choices that need to be made in this area. In terms of regulation, the level of the FDA and also other international regulatory bodies, there are several concerns. One of them that's been voiced a number of times by investigators is that there's, in general, a focus by investigators and industry on the use of already FDA-approved or easy-to-approve approaches for materials such as the stem cells and matrices that might be involved. The idea being that you get rapid FDA approval, get your IND, and get your pre-market approval, and then get out into the market ahead of the other people with something that could be used. The problem with this is that taking that approach versus going for maybe a better cell source or better matrices down the line, which is going to take a long period of time, is that you're going to end up with, again, this mismatch with incremental improvements versus breakthroughs and really a killer app for stem cell use, and that you run the risk of souring the public on stem cell therapies, common problems with the regulators, if they're not seeing really useful therapies coming down the line, and also with investors in terms of moving this forward into the clinic. And then we're also dealing with issues in terms of regulation of clinics that are doing stem cell therapies at this point and are potentially going to really cause a problem with the public's perception of the legitimacy of stem cell therapy. And this comes back again to a major problem that we're facing in the U.S. and I think also in Europe and other countries is very long and complex regulatory paths that the regulatory agencies really aren't set up to handle stem cells. They're really thinking in terms of older technologies like pharmaceuticals and first-generation biologics. And so I think the FDA needs to have increased flexibility, speed, and open interagency and interagency communication in terms of developing rules and policies and in processing of these applications. Right now, a lot of this is a case-by-case basis and is very difficult. So what might we want to target? What disease states or injuries should we really be going after that would be the best to initially use for stem cells and maybe have proof of concept? And this is a difficult area. I think that there may be early and late-stage targets. Right now, I think the U.S. is focused in large part on what would be the easiest to do first. And I think what you're seeing in terms of the U.S. clinical trials is its focus on bone fusion, particularly spinal fusion. And this is an area where you're looking at procedures that are already commonly done with allogenic grafts and with autologous whole bone marrow. So moving into using of stem cells is really problematic in that most of these are being used as stem cells being used are mesenchymal stem cells, either from bone marrow or from adipose tissue, in that this may only have a really marginal improvement over the use of autologous whole bone marrow in the current therapies that are in place. But this is something that may be easier to get through because it's something that is closer to what's already approved. Another thing to think about is where would you have the biggest impact, where the largest number of potential uses for stem cells in orthopedics, and where you might also then help drive this by having higher clinical and investor income. And so areas that I'll talk about in a minute include fractures, joint replacement, allografts, osteoarthritis, cartilage repair, osteoporosis, and various surgical procedures. And this seems to be an early focus internationally more than in the U.S. Also, could there be a breakthrough area that showing that you're getting really strong benefit from the use of stem cells versus just incremental treatments? And this is the second area where the U.S. is actually focused, where we're looking at the use of stem cells in osteogenesis imperfecta. And this could potentially be a big benefit where you have a ability to have a major impact on the disease for the patient. Also, where is federal dollars pushing things? In the U.S., there's a big push from the Department of Defense for trauma injury repair and the Armed Forces Institute of Regenerative Medicine has a very nice, innovative program that Todd McDevitt might be able to talk more about that is focused on getting innovative therapies using a business model out there as fast as possible. The one concern that I have with this is that as we're winding down in the Iraq and Afghanistan wars, that the support for this may dwindle eventually. But right now, I think this is a very innovative area, pushing forward innovative science and clinical approaches with stem cells. And I'll talk more about acute injury in versus chronic in a second. If we look at potential areas of orthopedic utility for stem cells, various surgeries like orthopedic knee, hip, and spinal surgeries where you could use them to enhance prosthetic engraftment, anchorage, and healing of the surgeries, as well as bone fusion, are potential areas. Orthopedic complex fracture repair, non-union bone gaps. Again, there's a number of civilian and military drives for this. Orthopedic simple fractures. This is actually an interesting area where you may have the ability to use increased speed of repair and may get spotlighted and highlighted with sports injuries where professionals in collegiate sports could help highlight the potential for this in shortening the time and return to either duties or to improve the patient outcomes over a shorter period of time and have a better prognosis. Oral and plastic surgeries are areas that have also been looked at: jaw repair, facial bone reconstruction, and particularly following oncological surgeries. Genetic bone disease, again, osteogenesis imperfecta, osteodysplasias, and potentially also osteoporosis therapies and rheumatoid arthritis. If you look at the numbers, there are very large numbers in the U.S. in terms of these surgeries. So total hip replacement is over 250,000 done a year. Knee surgeries over 600,000 done. Spinal fusions almost 300,000 done a year. Again, this is an area I think that the U.S. is pushing forward initially. Osteoporosis has a potentially very large patient population. Vertebral compression fractures, there are about 700,000 a year in the U.S., as well as other fractures, hips and wrists. 44 million individuals in the U.S. have osteoporosis, so this is a very large market. Bone fractures, there's 6.8 million fractures in the U.S. a year. And allograft transplants, there's over 1.5 million allografts, and this includes those that are used in spinal fusion. Oral surgeries are much smaller numbers, in the tens of thousands, and obviously genetic diseases are much rarer. For example, osteogenesis imperfecta, there's only 20 or 50,000 people in the U.S. who have survived with this disease. So what is the cell sources that we should be using? Well, the first big question, I think, is allogenic or autologous. And this is an area that has had major debate among investigators, clinicians, and companies. And ultimately, I think autologous looks best, but I think there's uses for allogenic. The biggest issue is immunological, whether or not there's going to be rejection by the patient or graft-host disorders. I think it's going to have an easier regulatory pathway. The cons are the need to expand autologous cells and the regulatory issues that will be dealt with on that. And that in order to do this, since it will become, in effect, a personalized medicine, each patient will need their own stem cells expanded and possibly modified, put in a matrix, and put back in. We need improvements in the technology to do this, to make this effective. Allogenic obviously has major value in terms of off-the-shelf, being immediately available. It lends itself better to manufacturing, intellectual property, and commercialization, may be easier to develop in the short term. Again, the biggest issues here have to do with immunosuppression and potential effect on healing, whether we have a lifetime treatment, although this may be moderated by the fact that you may not need to have the stem cells in place for the patient's lifetime integrated into the tissue. It may only be needed for a short period of time during healing repair processes. So this may still have value in doing that. Another method that might be used for allogenic cells, if they're going to be engrafted in the patient for their lifetime, is HLA haplotype matching and banking. It's been estimated that 30 cell lines could match 80% of the Japanese population. 100 cell lines could match the world, around the world could match the majority of patients there. So where should we get the cells in terms of which tissues? Embryonic stem cells are obviously an area of great interest. The biggest advantage is unlimited expansion and pluripotency. The pluripotency is a problem in terms of tumorigenicity, and so there's been a real push to differentiate the cells to a more limited potential, such as MSCs. Again, though, we were facing the problem of having to be able to pull out potentially tumorigenic cells from this population prior to use. Again, they're allogenic. All the issues related to immunosuppression are problematic here. Fetal adult cells would include those such as umbilical cord cells, amniotic fluid cells, adult somatic cells, we're going to talk about a little more in a second, bone marrow stem cells, particularly derived from mesenchymal cells, or small embryonic-like stem cells such as MSC cells. Actis is marking these in terms of multi-stem, or possibly committed pre-osteoblasts might be useful. Adipose-derived cells is a large number of these cell types that are well-defined: stromal cells, parasites, and MSCs. Again, induced pluripotent stem cells may have utility. So with umbilical cords, again, pros are multipotent. They're non-embryonic. Cons are these are typically rare cells, very small numbers to begin with. They're going to have to be amplified. And if they're used allogenically, they're going to be the graft-host issues as well. Possibly cord blood banking would be a way of going around this. Very useful potentially, though, autologously, but this would probably be fairly rare since you'd have to bank this well in advance. The biggest source of cells that are being used currently and probably are going to be used in the future would be adult stem cells. Again, no ethical issues. You can use them autologously. You can maybe use them as matched or unmatched allogenic as well. MSCs are very interesting in that they are immunotolerant and immunotolerizing, and therefore, while their MSCs may avoid inducing rejection by the host, however, once the cells differentiate, there could be problems in terms of them expressing immunogenic markers and being removed. So long-term engraftment MSCs that are allogenic may be an issue, but again, they may not need to be engrafted long-term. Interestingly, parasites, Bruno Pinalt has brought up this as a possible stem cell source out of adipose tissue, may be obtained in very high numbers, such as 10 million parasites out of a liter of fat, so they may not need to be expanded and can be potentially used autologously as a non-expanded population. They are interesting also, they engraft better than MSCs do into the bone marrow, and they are potentially more precisely characterized because of their cell markers. And again, the pros with adult cells is multimodal effects, but paracrine, which is probably a very important thing in terms of tissue healing and repair, as well as cell differentiation, which is more important in terms of long-term structural repair. Cons, again, obviously limited potency. Problems with defining MSCs and adipose-derived cells, very heterogeneous populations, and this is an area that needs improvement. Limited numbers of MSCs, and we'll probably have to grow and expand them to be used. And as I said, these are already being used in therapies, principally as autologous cells. This is a very commercial, potentially valuable area using allogenic MSCs, and this is an area that Athersys and Osiris are already using and planning to use for orthopedic surgeries. IPS cells, again, have issues that are very similar to embryonic stem cells, but they have some additional value in that they could be autologous cells and therefore avoid the immunological or ethical issues and even be used in gene therapy. Another area that's of growing interest to stem cell investigators therapeutically is the use of endogenous cells, that is, cells that we would not expand ex vivo but would expand in vivo, and then could mobilize and target to disease and injury sites. And this is an area that my lab is very interested in. You can manipulate the microenvironment the cells are in to induce proliferation and then migration or mobilization and direct the cells to osteogenic surfaces. It's a very interesting article just came out by Guan et al. Using a synthetic integrin ligand that would bind to MSC integrin receptors, activate the cells, and then have another modification to target them to the bone surface where there's active bone formation then. So this is an area of growing interest. Also, you have the potential for implanting scaffolds that then become populated by the endogenous cells by using technologies to drive the cells, again, proliferate them, mobilize them, and then target them to the scaffolds. So matrices, this is an area that we don't really have time to go into in huge detail, but is, I think, a major area of interest, and I can't do justice to it today, and hopefully some of our panelists can address some of these issues. It's turning out, though, that stem cell engraftment, particularly long-term engraftment, is much improved when cells are embedded within a matrix. And this is also the whole issue of tissue engineering. The idea that we can also use novel matrices instead of just simply demineralized bone or acellular collagen, but we use these synthetic novel matrices that provide new approaches, self-assembling matrices, superphysiological matrices that drive bone repair maybe more than a natural matrix would or a biomimetic matrix. There's a concern here that the focus on the FDA is to try to use natural matrices or already approved matrices, and that we'll miss out on the potential for having real improvement, not just marginal improvement, if we sort of don't use these newer matrices. Again, it's obvious that osteogenic potential is driven by both the matrix the cells are in and the microenvironment the cells are in terms of soluble factors as well. And there's a great push right now to look at what's the appropriate 3D microenvironment for the cell and how to construct it. And there's a balance right now in the literature in looking at the degree of rigidity, for instance, of the extracellular matrix and the degradability in terms of driving the phenotype of the cell. And that particular cell types that we want to have differentiating to particular cell phenotypes, we may need to customize the matrix for those cells. And also creating niches for stem cell engraftment in terms of having a stem cell niche to add to that population to help drive repair possibly for osteoporosis would be of interest. Okay, so I think some of the challenges that we're facing technologically here also are important to look at, and this includes the technologies to isolate stem cell populations, to identify particular subpopulations and acquire the right cells that you want to use in your therapy. And this is an area where there's a lot of interest and movement right now in terms of identification of markers and isolation approaches. So the technology to isolate these cells, particularly if we're going to do autologous stem cell repair, is important. So we can do this quickly and meet regulatory approval. Another issue is scalability of the cells, particularly if we're going to grow up cells in large enough numbers to be used. There's an interest in bioreactors and bioprocessing systems that would allow us to retain a very specific phenotype or pluripotency of the cell, and one that, in terms of regulatory requirements, is very repeatable and consistent in the cells that are being grown and differentiated. Construction technologies of matrices are an area of active interest, and I think it's very important to get the technologies here to scale things and make the scaffolds and the microenvironments to optimize cell implantation and function, and do so in a way that's going to be economical, because we have to think back on all these approaches, what's going to be fundable by insurance, what's going to be fundable out of the patient's own financial abilities, what's going to actually be something that will be pragmatic in the real world of healthcare. Modulation of the microenvironment cells to direct them to specific phenotypes, again, is, I think the technologies is an important area for growth here, and there's a number of challenges here. I think we also need to consider the transition in terms of the biomechanical properties, having either adaptable or tunable matrices, because when we implant the cells and are going for growth and repair initially, that may be different of what the matrix needs to be once the repair is finished and the tissue is now being used in normal daily life. So I think the technologies of degradable or tunable matrices is critical to be developed here as well. So if we think now about how some of the regulatory issues may affect some of our choices, in the U.S. right now, all stem cell therapies have to go through the FDA, and within the FDA, they're going through the Centers of Biologics Evaluation and Research, CBER, first. This is not the only area in the FDA that's going to regulate stem cells, but right now, the policy is that most of these stem cell therapies will will go here first. The INDs and the IDEs will go here first. And now we have to have a distinction. Is the cell therapy they're using one that is under the category of human cells, tissues, and tissue-based products, HCTPs, which falls under a particular section of the regulations called 351, and that's going to regulate biological and device and drugs. And this is an interesting area because these three categories, biologic, device, and drug, also have three separate centers for regulation. So devices are under the Centers for Devices and Radiological Health, and if it involves a matrix, this group will probably also be involved to some degree. If it's a drug, it's going to go under Center for Drug Evaluation and Research. So if the cell has been modified to express certain molecules, it may be considered a drug as opposed to a biologic. And a lot of the stem cell therapies are going to be considered combination products, which would be biologic and device or biologic and drug. So right now, as I said, everything is going through CBER, but that may not be the case in the future. And right now, everything is being determined on a case-by-case basis, and if it's not clear where it should go, then it goes to the tissue reference group with a decision as to what policy pathway, regulatory pathway it will have to go through. Now, the big, again, barrier for moving things into the clinic is the FDA regulations in terms of safety, purity, potency, effectiveness, manufacturing controls, consistency of lot to lot, being able to identify your cell product from other cell products, and stability. Each of these has very high hurdles to overcome and major issues in terms of getting through the FDA. Unfortunately, right now, this is still a very open field, and the FDA doesn't really know what to do in a lot of cases, and these things are being determined on a case-by-case basis with input back and forth between the FDA and the investigator, which is good on the one hand, but it takes a long period of time to get through this. Other types of cells would be considered minimally manipulated cell products and fall under Section 361, the tissue rules, and this is much simpler to use. You don't have to go through a clinical trial. You don't have to prove efficacy. You just have to show that these cells are going to be derived from a patient and then used by that patient again autologously or within a close relative, that the cells are not going to be expanded in culture and they're not going to be exposed to drugs. So if you basically take out a patient's MSCs and isolate them, you can put them back into the patient without FDA regulation. I shouldn't say without FDA regulation. It's still subject to licensing and manufacturing concerns and making sure there's no contamination of the product, but you don't have to prove efficacy. You don't have to go through the whole process of the applications. You don't need the INDs or IDEs or pre-market approvals. You can go straight to market. And the potential then to kind of avoid the 351 rules by going with autologous transplants of cells that you don't expand. And this might be possible to use, for example, with parasites or adipose-derived MSCs, and then use them in the patient. But the reality is that we're going to most likely want to add matrix components or modify the cells in some way by exposure to cytokines prior to use. But now most clinics in the U.S. are operating under this 361 rule, and so we're seeing therapies, again, that are not based on clinical trials that are being used. In the U.S. right now, there's a clinic in Colorado that has a lawsuit against the FDA which shut down their one of their therapies, which was taking bone marrow-derived MSCs from a patient, expanding them, and then putting them back in the patient. The FDA said because they were expanding them, they then fell under the 361 rules, that they were HCTPs, and moreover, they said that they were a drug, and this has caused a lot of confusion and problems with the FDA considering them a drug, since that's going to go through a very different type of protocol for getting a policy approval. So this lawsuit's in place right now, and the FDA is not talking about it, and there's some issues related to what type of cells may fall under the 361 rules versus the 351 rules. But the outshot of this is that we now have these unapproved but permitted therapies that are getting ahead of approved therapies, and I think this should be a concern to stem cell investigators as well as the FDA in terms of what these clinics might be able to change public opinion in terms of the value of stem cells. I'm going to stop here and post the questions I initially had, and let's go ahead and have the panel come in.
Well, thank you very much, Dr. Hill, for this presentation. I think you perfectly illustrated the great complexity of this field, both from a biological point of view, with the large possibilities of choosing the cell types and sources, and of course the very large complexity of the regulatory issues. Before we go to any specific questions, there is really one point I would like you and the people from the panel to clarify for me, because from the point of view of the potential applications of this technology, it seems to me there is on one side the potential for bone repair and regeneration in areas that are where the defect is focused, like spinal fusions or non-unions. And I understand well that maybe through use of scaffold, that we'll discuss further, this can be achieved by any sorts of stem cells. Now, on another side, there is also, and you mentioned at the beginning, some hope for a more systemic regeneration or whatever, rejuvenation or repair of systemic skeletal disorders, such as OI and maybe even osteoporosis. And I'd like to understand what is the evidence we have so far that indeed the injection of mesenchymal stem cells or other stem cells can achieve a systemic effect on the skeleton rather than a local effect. So I don't know who would like to first answer that first question.
Serge, this is Sandeep. I'd be happy to try to address that. And I also wanted to thank Dr. Hill for really a terrific presentation. So, you know, I come from this from a perspective of a clinical investigator in osteoporosis research and a clinician in osteoporosis. So obviously the big disease is osteoporosis. And, you know, one of the questions was, you know, would these kinds of approaches be applicable to those kinds of systemic diseases? So even setting aside osteogenesis imperfecta for now. And I guess I have some skepticism that for those for osteoporosis in particular, that this is going to be a competitive approach as opposed to small molecule or other peptide therapies. And it also gets to a broader question, I think, that the whole stem cell field is I think, grappling with. And if you talk to leaders that head up stem cell institutes at different institutions, they sometimes have very different perspectives on this. And there's kind of the cell-based therapy camp versus, you know, using cells for in vitro studies for high-throughput drug screening and so forth to develop small molecules that then work on endogenous stem cells. And I guess for osteoporosis, I think probably the latter approach, you know, whether you use MSCs or IPS cells or other cells to really develop additional pathways that could be regulated through some kind of high-throughput drug screening approach. And potentially that's also applicable to osteogenesis imperfecta or other diseases where you have IPS cells and can kind of study them in vitro, because that approach does get around a lot of the regulatory issues that Dr. Hill brought up. And that contrasts perhaps with more local therapies where the cell-based approaches may be much more viable. So that's just one perspective. I'd be interested in you know, Dr. Hill's thoughts or those of others.
Yeah, thank you, Sandeep. Dr. Kassem, let me ask to expand on that question, and I think you've investigated into the underlying question, which is, you know, is osteoporosis a failure of our mesenchymal stem cells in the bone marrow environment to regenerate bone systemically? And in that case, could this be circumvented by giving back mesenchymal stem cells?
I would like also to start by thanking Dr. Hill really for a wonderful presentation, and also for Sandeep for initiating this discussion. Actually, what I would like to start with, just to say that there are actually two approaches right now that are being envisaged to use stem cells in therapy. What is called cell replacement therapy, and I think Dr. Hill presented that. But there is another approach, which is a humoral therapy. And the background for the humoral therapy is that in a lot of studies in the non-bone field in particular, there are some therapeutic effects are as the result of really not the cells that differentiate into particular cell type, but by their ability to produce a number of regeneration promoting molecules, immunomodulatory molecules, inflammation modulation molecules. And actually this is quite an interesting area of research, and they can link with the comments of Sandeep. So I think we need to keep the perspective that there are two approaches in the use of stem cell for therapy. I do agree with Sandeep that the use of stem cells in the regeneration of the skeleton of an osteoporotic patient want to be on the top of my clinical priorities. I think if we make some clinical priorities, I would say that local therapy, if we are envisaging some local repair and regeneration, and here we can discuss the issues relating using cells combined with matrices to repair localized defects and local regeneration. And I think the other area where we envisaging a systemic therapy, then we need really to use the stem cell for their humoral properties and what they secrete, which is important for tissue regeneration.
But in that case, the idea would be to still inject the mesenchymal stem cells, maybe primed in vitro by some stimulus in order for them to produce growth factors and other things, or is it to use those cells in vitro, isolate those factors, and then work on this more pharmacological approach from what we've learned from mesenchymal stem cells? What are you alluding to?
I think I do agree with you. We don't have a strong evidence, actually, that if you inject the cells, you will get a huge therapeutic effect vis-a-vis bone formation or systemic regeneration of the skeleton. There are some studies in the literature, but they contain a lot of confounders, so we are not sure really that the effects, observed effects, are due to the cells injected. But even in my research, I advocate for trying to isolate these particular cells, these particular factors, and try either to use them or factors and the pathways that are important for the endogenous regeneration of skeletal stem cell, and use this factor for systemic therapy. I really, right now, just injecting the cells systematically and trying to study and try to examine the regenerative potential. I think it's a very early stage, and it should be done in a large number of preclinical trials to be sure how to improve the homing, engraftment, and the efficacy of the functionality of the cells. So it's a long way really to.
Well, I agree with all these points. One of the real issues with osteoporosis is the loss of the stem cells with age and disease. In trying to replace them is very technically challenging to get the MSCs to go to the bone marrow in the first place and then engraft. And the second is once they're engrafted in this diseased environment, and the evidence is that they're not going to be able to correct that environment on their own. So you would have to modify the microenvironment for the cells to make the cells more osteogenic and in fact, repair the disease process. So I think in the short term, this is not something that's going to work out. We need much more work on the development of approaches to get the cells to target to the bone marrow and to change the bone marrow microenvironment for the cells to act appropriately.
But this approach of systemic delivery was already used for heart disease, and I think that was not a really big success. So it's very difficult for the cell to go where you want them to go. And so for the bone, I think this is really questionable if this is working or not.
I agree.
Okay, well then let's maybe put in parentheses and stop our discussion about the systemic effects of mesenchymal stem cells here and focus back on what we could achieve at a more local level in spinal fusion or fracture repair, non-unions, and those problems. Now, Dr. Pioletti, you've worked on those cells and done some engineering of scaffolds to use those cells. I'd like to have your opinion on where we are in that particular area of orthopedic research, and also maybe I'd like you to comment on which type of mesenchymal stem cells, and do we need mesenchymal stem cells? Why not use osteoblasts, like the way, in the same way we use some chondrocytes to cartilage repair?
Yeah, actually that was exactly one of my points, is the reason for using stem cell in bone could be some way questionable also, because it really depends what you want. Do you want them to produce growth factors so they initiate the cells of the host to produce something, or do you want them to produce the extracellular matrix? And so I wonder if stem cell could do this work by themselves, or as you mentioned, if we can use, because for cartilage it seems to be more successful to use directly the chondrocyte and not the stem cell. And so that was one of my questions to Professor Hill. What is your opinion on using directly a differentiated cell, osteoblast, instead of stem cell?
Well, I think that may actually have a lot of utility. And as I said, there was an interesting paper that's just come out from Nancy Lane's group, where they've used these ligands to direct MSCs and probably osteoblasts to the surface of bone and help drive new bone formation. So the idea of putting osteoblasts in may be very useful, since they're going to have the right markers to identify the ECM in the areas for bone formation, whereas the MSCs don't initially and have to be driven to become pre-osteoblasts and osteoblasts. So this actually, I think, has a lot of utility.
Dr. Kodno, you are a basic biologist that has worked a lot on these basic mechanisms by which mesenchymal stem cells can regenerate tissue. So I would like to hear from you. What's the best approach? Allogenic, autologous, derived from the bone marrow, from adipocytes, from other tissues, MSCs, or more differentiated cells? Could you please comment on these aspects?
Hello, everybody. So yes, definitely. One thing that we can say first is that bone is a tissue that has very high regenerative capacity, and also Dr. Hill said that NIH supports basic research rather than translational research. Surprisingly, we still don't fully understand and have a very limited understanding of the basic mechanism of this high regenerative capacity. So what do we know about this process, and what can it tell us about the information we have and help us improve these cell-based approaches? So there's 3 points I would like to make is what does it tell us about the cell sources that we could choose, the method of delivery, local versus systemic, or whether we should use stem cell versus differentiated cells. So regarding the source of cells, what we have recently learned about the cells that participate in the normal repair process, thanks to a lineage analysis approach in genetic mouse model, is that cells seem to be recruited mostly locally and not recruited systemically, and in particular from the bone and not the bone marrow, but most importantly from the periosteum. So it tells us definitely that maybe certain cell sources might have have better intrinsic properties to repair bone. However, MSC derived from bone marrow are a lot better characterized. So I think that by understanding the characteristic of periosteum-derived cells and the factor that supports their regenerative capacity may help us better drive mesenchymal stem cell after transplantation. The other point is local versus systemic. We know that cells endogenously are recruited mostly locally. So in a local delivery approach may appear more intuitive, and particularly for bone fracture and defect. But as you said, other disease of the whole skeleton may require a systemic approach, and here we need to definitely improve the homing capacity of the cells. And the last point I would like to make regarding the choice of stem cell versus differentiated cells is that we forget when we talk about a stem cell, their definition is that they're able to proliferate and give rise to many different cell types after differentiation, but also their ability to self-renew. And a lot of approaches that have used differentiated cells or local transplantation of MSC that have shown the ability of these cells to differentiate and form bone actually show a transient formation of bone and not a long-term integration. And I think this is a real challenge to be able not only to drive those cells to form bone, but form bone that will sustain and not being resolved. Therefore, I think that an approach using stem cell might be more appropriate in order for those transplanted cells not only to differentiate into osteoblasts or chondrocytes, but also to go to the stem cell niche and in order to self-renew and allow long-term bone formation. And we really know so little about the skeletal stem cell niche that there's still a lot of advances needed here in our ability to understand the endogenous mechanism. So I guess, you know, my general comment about this whole discussion is that we definitely need these translational type of approaches that I think are crucial to define the condition to expand cells, manipulate them in vitro, and develop preclinical animal models. But I think the progress will come from parallel effort between different disciplines to improve these approaches.
Well, thank you. That actually sends us directly to you, Dr. McDevitt, and the question becomes whether on how the scaffolds can actually improve the survival activity performance of mesenchymal stem cells.
Yeah, so obviously there's been a lot of research for some time for cells, various types of materials as serving as cell carriers. And over time what has been pretty well established is that certainly will help with, first of all, upon immediate delivery, retention of the cells. So simply at most types of delivery or injection sites, it'll just help to at least locally concentrate the cells and keep them there by just providing sort of the physical attachment and the physical void filling space of many types of, especially defects, but even small fractures or cracks, things like that. The second part of that, though, is as we know more about the cell biology, especially, that's really been informative in coming up with more molecularly designed materials. And so there, the intent has been to introduce, starting with the adhesive ligands that are capable of interacting with cells, but then those can also in turn promote specific survival signals to the cells and instructive signals even in terms of differentiation. And now it's gotten even to the level, a lot of interest in recent years about the other physical parameters associated with the materials. And nice work I know from a colleague at Stanford, Sarah Heilshorn, shown that even just the acute survival of the cells can be enhanced by using viscous types of materials because of basically the shear forces introduced when you inject something through a needle. So for those types of delivery approaches where it's going to be maybe catheter-based, for the scaffold-based ones, though, also the mechanics of the scaffold itself, some of that underlying support that can play a permissive kind of role. It may not in and of itself be as potent as some of the biochemical signals. Pretty clear from various types of studies, largely starting in vitro, but now there's increased ones moving to in vivo environments where the local mechanics, both of the scaffold itself and the environment the cells are being exposed to, can also have some impact on the phenotype. So there's a lot of parameters. The biggest challenge in those is how do you take all of those considerations into account when you're trying to design for something, or how can you effectively control those, because oftentimes a number of those parameters, things like the permeability of something also affects its mechanical stability, degradation over time, those parameters. So they're all interrelated, and so it makes it difficult, at least a priori, to come up with, say, the material for even a single type application or knowing how to vary those. So there's still a lot of need, I think, in that kind of area to really come up with scaffold or material-based design considerations that are appropriate for the different types of wound environments which the cells may be introduced.
Thank you, Dr. Pioletti. You also have worked on scaffold. You want to comment further on the utility or the added value of those scaffolds toward the lifespan and viability of the cells?
Yeah, actually, in my point of view, there are two aspects with the scaffold. First, if you want to use the technology of stem cell In orthopedic, you need first to make sure that there is a functionality of what you're doing. So the scaffold obviously can bring this functionality from a mechanical point of view. And also, we recently showed that by combining mechanical loading on scaffold in vivo situation, you can increase the bone formation when you have, in our case, we also use the fetal cells. So the scaffold can really do two things. Obviously, as I said, to stabilize from a mechanical point of view if you have a fracture, for example, but also to help to transmit the mechanical loading to help the cell to produce more bone. So in my point of view, most of the time you need a scaffold, and that's also a point I would like to raise. Where do you need only cell in orthopedic situation? And it is not very often that you need only cell, you need the mechanical support. So by combining both, obviously, you will have something very interesting.
Yeah, that's really interesting perspective. I think that the mechanical stimulation of in the environment of the MSC through the scaffold could further enhance the biology of those cells. Dr. Kassem, I'd like to. I know you have to leave soon, so I'd like to hand it back to you and ask you what is your view at this point on which type of cell and which approach we should use.
I think actually maybe also because you can see there are enormous variety of the approaches one can start with, but maybe if we try to classify what we have right now in terms of the clinical applicability, so I would say that the cells that are ready to, and they are actually being utilized in clinical trial, this will be bone marrow-derived skeletal or MSCs. And the reason for that is actually these cells, they have the most proven bone forming capacity. The adipose tissue-derived, the MSCs, is still debated their functionality with respect to bone formation potential. And actually sometimes when they are used, the positive effects most probably are due to the humoral factor that I alluded to previously. So I would say bone marrow-derived MSCs to be used in treatment of localized defect plus and minus scaffolds. And the scaffolds here, you've heard also, it can be either a scaffold by themselves or actually, which is a very interesting approach, is what's called functionalized scaffolds. And this is a scaffold combined with peptides, or actually some also very fascinating approaches combining scaffolds with siRNA or microRNA, which is actually opened up an enormous potential. And also for testing, as you asked us, what is the current evidence? How can we prove that these cells really, they are differentiated, they form bone locally? And I think some of these functionalized scaffolds will allow us to, when they are combined with the new imaging modalities, to prove that the transplanted cells, they do what we expect them to do, which is bone formation.
Thank you. Well, we are getting towards the end of this webinar, but before we close, I'd like to sort of go back to Dr. Hill and maybe Sandy Kansla as well, to ask you then sort of to wrap up here. So at this stage where we are in this research, what is the most clear rate limiting step, and where is this field going, or rather where should it go so that we can really achieve some success in the development of this technology? So what's the rate limiting step at this point, and what are the perspectives? Dr. Hill first.
Well, I think in terms of developing a therapy that can be used broadly in the clinic and is shown to be effective, I think getting it through the FDA is one rate limiting step. But I think the cell types that you're using are critical. I think that the focus on being able to use autologous cells and developing the technologies to be able to amplify those cells and expand them, and then to be able to target them on matrices to where they need to be, I think are the most important ones that I would focus on.
Thank you, Dr. Hill. Sandeep Khosla, what should we do to move this on in your view, and where is the major problem we have now?
Yeah. Well, I think Dr. Hill, touched on one of these, and I think this field really is at a point where if these kind of unproven therapies that are being marketed all over the place kind of keep going, it has the potential, as Dr. Hill mentioned, to sort of color the whole field with a series of failures, basically, because all these unproven therapies are going to be out there. So I think that's a real challenge for the FDA, the scientific community, to maintain the credibility of the field, in contrast to these these small clinics that are starting to do some of this. So I think that's a huge challenge. I think the other issues have been summarized nicely. One other point that I'd just like to bring up that we haven't touched on is, just more about the fundamental biology. So, I mean, hematopoietic stem cells, for example, have been used in transplants for, a long time, and we know they stay around for life. And we don't really understand why that doesn't happen with the mesenchymal cells, whether they're given locally or systemically, why they eventually kind of disappear over the course of months and so forth. So understanding, that biology further, and some of that obviously is going to be influenced by the scaffold and so forth, but a lot of it may be cell autonomous, and understanding those questions is really going to be helpful.
I agree.
Well, I think that is an excellent conclusion to this webinar, and therefore I'd like to stop it here. To thank again our panel and our presenter tonight for a great discussion about this very complex topic, to thank our numerous audience tonight, and to wish you all a good day or a good night wherever you are, and of course to invite you back soon for another webinar on bone heal. Thank you very much, and
thank you.
Thanks.
Thank you.
Thanks. Bye.
Thank you.